Bismillah ar-Rahman ar-Rahim. As suggested by last lectures, I open my channels so I can present my lectures through the YouTubes, and this is not enough for discussion, but you just can add it advance to the e-learning. So please, if you have any questions regarding these lectures, you can send it to me through my email or by WhatsApp or online. Even we can use the Facebook sometimes. Yeah, this first lectures on YouTube about the amino acid protein structure and function. We generally, as I told you, we use the reference living code, for example, chapter 2, page 13. Yes, what are protein? And we told that protein are polymer of amino acid joined together by peptide bond. We already covered this part in the last two lectures, so I just will go through uh, about proteins, function of the protein, and I told you that protein is the most abundant functioning diverse molecule in living systems, and they do so many functions. One of the functions of that protein, I told you that enzymes, for example, and hormone regulate metabolism and use as a framework like collagen, and use transport nutrient like albumin, and immunoglobulin used for defense. Sometimes we can use protein as energy, but this is not the main function of the protein. Protein is the main for the building the body framework. So that's, and I already asked you in the last lecture what is the difference between the enzymes and hormone. Please do it as a homework. I told you there's more than 30 difference between the enzymes and hormone, and give example. It's okay. And that's where we continue about the protein, the function of the protein, and we, as I told you, protein are the polymer of amino acids. They consist of 20 amino acids. That's very important, amino acid. But there's more than 300 amino acids in the nature, but only 20 amino acids used for protein synthesis, and that's very important. And I told about the peptide bond that join together amino acid and another amino acid. Breakdown of the peptide bonds is difficult, but sometimes they can. When we use a strong acid or base, we can break down the peptide bond. And that is how to join the peptide bond between two amino acids. Already I talked about this. It's just revision. And when you go for amino acids classification, I told you we classify in different according to, for example, uh, attached group, carboxylic group, amino group, side chain, polar, non-polar, and we told that only proline have amino acids, exceptional, and glycine is the smallest amino acids. And this we talk about the isomers. We go for that L amino acids is most abundant and not the D1. This is about the 20 amino acids classification. Yes, acid base, polar, non-polar, essential, non-essential, amino acid. Please always revise and recall the knowledge about the, when we talk about the essential, you should list figure out the essential amino acid, non-essential amino acids, ketogenic, glycogenic amino acids, amino acids that contain aromatic compound, and all of this you should be familiar with the classification of amino acids. We just go through. Yes, when we talk about the sickle cell disease, we just say that single change of one amino acid they can change the nature of the protein. For example, hemoglobin S, only glutamate change it to the valine, and this makes the sickle cell disease. And it's a common, as I told you, in Eastern Sudan, in the west, I think so, not western, in the western region, Darfur, Kurdistan. Also, it's a common in Saudi Arabia, Mediterranean, sickle cell disease. and we just go down about the pK value. And I told you what's the difference between the pK and the pH in the last lecture. You should be familiar as a pK or dissociation constant, the pH at which 50% of the acid is dissociated. pH is a minus log of hydrogen ion concentration. And always remember, the smaller the pK, the stronger the acid, the larger the pK, the weak acid. The maximum buffering capacity occurs at the pH pK range plus or minus 1. Yeah, and we talk about the buffer in the last lecture. We said the buffer solution is the resistance, the change in the pH. And I told you, please mention some buffer system in the human, for example, which is a buffer like hemoglobin and carbonic hydrates that use bicarbonate, for example, they can use for buffering systems. So amino acid act as a both acid and base, has amphoteric properties. So they can act as acidic, they can act as a base. This is the nature of the amino acids. And this we talk about the Zwitter ions, Pi, BH, which is a molecule is electrically neutral, and we talk isoelectric point, and we talk about the pH. Most aminos contain both positive and negative charge with a net charge of 0. And we give some example for amino acid. We talk about the gamma amino acids. As I told you, this is just a revision for the last lectures. And we talk of the some neurotransmitter as a gamma histidine is the mediator for allergic reaction. We know sometimes when the cells the cells yeah, this is example for some compounds that derive chemical compounds that derive from the amino acids. For example, GABA. We know that GABA is a derivative of glutamic acid, and dopamine from tyrosine are neurotransmitters. It's very important. Is important neurotransmitters. Histamine, that is derived from histidine. We know it is very important for immune system. Allergy. Some people they have allergic reaction. They can use sometimes antihistidine, antihistamine, for inhibit the allergic reaction. And also the thyroxine, derived from tyrosine amino acid, is very important. We know that for the goiter. Yes, and we talk about some polymer of amino acids, how the peptide bond is joined chemically, and these some peptides. Besides the peptide bond, there is another bond we call it disulfide linkage. Some amino acids they contain sulfur compound, S compound, hybrid. So that means they can make sulfur linkage between two amines like cysteine units, for example. Cysteine and methionine they contain sulfur. This is how the amino acids they join to the peptide bond. Yes, that is how the cysteines is joined. Also, they make the linkage. And this is of the contoxin G1, how it's a peptide, polypeptides, how they join polypeptides, very important polypeptides. Also, glutathione, we know glutathione is very important for free radicals, and this consists of cytosine and glycine that has a numerous important function within the cells. Glutathione is very important for conjugated drugs to make them more water soluble. And we talk about the cofactor for enzymes, for example. These amino acids are already abbreviated by 3 letters. For example, if you say alanine, ALA, and there is abbreviation on 1 letter A. So please try memorize all of these names for amino acids, 20 amino acids, the 3 letters and 1 letter. This is very important for us using the 1 letter because we know that in the protein synthesis, the polymer, sometimes when we make protein synthesis, we just use the letters, single letters. For example, if we make sequence for DNA for protein, for example, insulin, and we know the sequence of nucleotides, AATCC, for example, and after that we transfer it to protein synthesis to amino acids, so we use the letters, first single letters, A, for example, A, R, N, D, and you should try to memorize this. Yeah, this is the first important. That's almost for me it's a revision. But here is my beginning of the lecture. We talk about the protein structure. Protein is very structure is very important. What are protein? I told you, protein have polymer of amino acid. There is the four level of structure: primary, secondary, tertiary, quaternary structure for protein. So this structure is very important for protein to do their function or activity. Protein folding and stability is very important using for protein structure. Protein denaturation also to lose their activity. So protein misfolding and disease, this should be covered in my lecture now. I just will go through all of these, the four levels. I will talk about the primary structure, I will talk about secondary structure, tertiary, quaternary structure, and I will go for the protein folding and stability, how we fold the protein. After protein synthesis, they have some modification to do their function, like folding and stability. And we talk about the denaturation of the protein. What is it? How can we done? How can we done? How can we make denaturation for protein? And we in the last we will talk about some misfolding and disease regarding the misfolding of protein. Structure and function relationship. What are protein? I already told about that. So let me go for hierarchical nature of protein structure. Protein is very important in our body. Protein, any defect for the protein structures, they can lose their function. So to do their function, protein, they have different structures, should be collected together to carry on the function. Any defect of this, they can lead to that disease. Primary structure, talk about the amino acid sequence. Talk only about the peptide bond sequence, single lines of amino acid. Secondary structure, I talk about alpha helix and beta sheets. Tertiary structure, we talk about three-dimensional, 3D. Quaternary structure formed by more poly chain units. So let me talk about the primary structure of the protein. As I told you, it is the sequence of amino acid in the polypeptide chain. It's the sequence of amino acids in the polypeptide chain. Understanding the structure of protein is important because many genetic diseases result in the protein with abnormal amino acid sequence. And we give example for sickle cell disease, how the glutamate gentotrophalin. So also in folding and loss of the impairment of normal function, and we will talk about some disease that have been due to the misfolding of protein. It helps diagnose or study the disease. When we do the primary structure, for example, we know the structure, for example, for insulin, any defect for any sequence, we can make a disease simply when there is a mutation of the sequence. So protein are linear polymer of amino acid primary structure, as I told you, contains the peptide bond. In the last one, if you look at it, ATF sequence, this is amino acid sequence. It is abbreviation for amino acids. So many amino acid linked together through the peptide bond and describes the prior. And this is the peptide bond. I talked it already about it. Secondary structure of protein. Here we are talking about the another structures like alpha helix and sheet, alpha sheet, coiling, folding, or binding of the polypeptide chain. They are two main regular form of secondary structure: alpha helix and beta plated sheets. Secondary structure, you are look at it, hydrogen bond between the alpha helix. It is almost look at the chain here. It is a alpha helix, right-handed and left-handed, and joined by hydrogen bond. Alpha helix is clearly like it's a helix, right bonded. It has 3.6 amino acid residue per turn, stabilized by hydrogen bonding between first carboxylic and fourth amino acids. So it's a amino acid already they join together by hydrogen bond. Beta sheets, usually two or more polypeptide chain, make hydrogen bonding with each other. Beta sheets are plated, as is in the structure. If you just look at double helix, two polypeptides, so they join like the sheet. It's antiparallel beta sheets. Hydrogen bonding is more stable. Antiparallel is more stable. Hydrogen bonding is less stable resorted في parallel beta. When they differ in the position of the sheets, they can make stabilizing the protein itself. Yes, that's how hydrogen bonding in the alpha helix and beta plated sheets. The difference between alpha helix and beta plated sheet. Alpha helix rod-like structure, coiled polypeptide chain arranged in spiral structure as a helix. Beta plated, it is a sheet-like structure composed of two or more polypeptide chain. Alpha helix, all hydrogen bonding are interchain, while in the beta plated, interchain between separate polypeptide chain and interchain in a single polypeptide chain folding back on itself. There is another difference, spiral or helix. Alpha helix is prevents the chain from being fully extended, but while in the plated they are already extended and relatively flat. The last one, it is abundant in hemoglobin Fg and myoglobin Ag. Example, alpha helix is abundant in hemoglobin and myoglobin, and we know that hemoglobin and myoglobin they carry oxygen. And beta plated is abundant in protein like cyclic fibrin and carbonic anhydrase. Carbonic anhydrase is very important enzyme for in the buffering. Other secondary structure, turn, first turn, loop, beta blended is not important, but we just focus on alpha helix and beta blended. This is example for fibrous protein. Fibrous protein provide mechanical support, often assembled into large cables or threads. The الخيط. Alpha keratin, major component of the hair and nails. Collagen is very important for tendons, skin, bone, and teeth. This is example for fibrous protein. Collagen is very important protein. It's a triple helix. Fibrous protein, a part of connective tissues, bone, teeth, cartilage, tendon, skins, blood vessel. Contains three left-handed coiled chain. The series repertain glycine. Glycine XY. X proline, Y hydroxyproline or hydroxylysine. So it's mainly glycine is very important component for the collagen. And glycine is essential amino acid and it's the simplest amino acids they contain H, COO, H, and NH2. It's a term, it makes a term, it's a fibrous protein, series residue term, glycine, XY, glycine, proline, hydroxyproline, or hydroxylysine. Glycine and proline in collagen, as I told you, is very important. Rich in glycine and proline amino acid. Proline facilitates helical conformation of each chain by causing kicks. Glycine, the smallest, fits into the restricted space where the three chain of helix come together. Non-standard amino acids, collagen, proline, convert. This is regarding the collagen. So please just go back, read about the collagen, how it's very important in the bone and teeth. Collagen disease is some disease regarding the collagen, scurvy, vitamin C deficiency is very important. Vitamin C is very important in the formation of the collagen itself. Hydroxyproline, hydroxylysine, these enzymes that they need vitamin C in the collagen formations. This is regarding the secondary structure as a fibrous protein, and when we go structure, tertiary structure of the protein, tertiary refers to the folding of the domain, are fundamental function and three-dimensional structure of polypeptides. The core of domain is is built from combination of super secondary structure. So it's a units tertiary structure already tell us the units. The following four type of interaction component stabilize the tertiary structure of globular protein. For example, disulfide bond, sulfide, they contain sulfur, hydrophobic interaction, hydrogen bond, and ionic interaction. This هذه روابط bond to stabilize the tertiary structures. You should look at this. This is a three-dimensional. So these all of these bond they make the three-dimensional units of protein. So domain three-dimensional structure polypeptide chain fold into two or more clusters known as the domain. Domain are functional units that look like globular protein. Domain are part of protein subunit. It's a subunit exactly, domain. It's very important. The domain is very important in the function of the protein, and that is examined in the globular protein. Globular protein usually water soluble, compact, roughly spherical, hydrophobic interfere, hydrophilic surface. Globular protein include enzymes, carrion, and regulatory protein. So when we go for quaternary structure of the protein, then that is the last for the structure of the protein. It is a process quaternary structure. When we just example for when we look at the hemoglobin, they have composed of subunits held together by non-covalent interaction, hydrogen bond, ionic, and hydrophobic interaction. It's just look at the hemoglobin, you will hear how it's folding in different subunits. Yes, that's it. Yes. Yeah, this just show for hemoglobin structure how it's used for units to do the function. Quaternary structure, many protein contain two or more polypeptide chain. Each chain forms three-dimensional structure called subunits. It's a 3D arrangement, different subunit of protein. The subunit may be structurally related or totally unrelated. Hemoglobin and myoglobin both can bind to oxygen. Both are globular protein. Myoglobin is only formed on only one subunit, في حين أنه hemoglobin is 4 subunits. A multi-subunit protein is called an oligomer. Hemoglobin is composed of alpha 2 beta subunits, 4 subunits. Forces that stabilize three quaternary protein structure, as we know it, hydrophobic effect, hydrogen bonding, electrostatic interaction, and disulfide bond. This can stabilize the protein. Disulfide bond, as you look at it, yes, sulfur, two sulfurs, SS, joined together. And we go for denaturation protein. Denaturation protein happen when they lose their different structure except the primary structure. بمعنى لأن you lose all the other structure, only primary stay. You cannot break the primary structure. So they just told you for you this sequence of amino acid polypeptide chain. Loss of native structure, biological activity of secondary, tertiary, and quaternary structure protein. It is unfolding of the protein. Denaturation agents include heat, organic, solvent, strong acid, or base. Also detergent, ions of heavy metals such as lead or mercury, liquor. That means when we denature protein, they lose its function. Protein denaturation, process which protein lost its function. Yes, native structures. Factors that causes heat, change pH, detergent. So changing of the pH is second change. This is how it's happening. It is a subunit. For example, if like hemoglobin, they become single peptides. Protein folding is very important, which occur within the cells in the second 2 minutes. Employ shortcuts through the measures all the folding possibilities. That means after protein synthesis, they make folding or translation to change it. That is as peptide fold, its amino acid chain are attracted and repelled according to their chemical properties. For example, positively and negatively charged side chain attack each other. Similarly charged side repel each other. In addition, interaction moving, hydrogen bonding, and this bond is very important for folding. All exert influence on the folding process. This process of trial or errors test many but not a possible configuration seeking a combines in which attraction of wet repulsion. بمعنى يعني yes, folding is very important. They need according to the bond. Sometimes ممكن يكون في misfolding of protein. Yes, protein misfolding. Every protein must fold to achieve normal structure conformation function. Abnormal folding protein leads to a number of diseases in human, like Alzheimer disease. Beta amyloid protein is made folded protein in the Alzheimer. It forms fibrous deposit on the plaque in the brain or leaked for the Alzheimer patients. That means some protein called beta amyloid is misfolding. Creutzfeldt Jakob or Parion disease. You know Parion disease? Parion protein is present in normal brain tissue. Some cow mud disease. You remember? They have a parion protein. In disease brain, the same protein is misfolded from alpha helix and beta sheet, therefore insoluble fibers segregate as the damaged brain cells. Protein misfolding. We continue about the errors process that sometimes results in the improperly folded molecule. These misfolding proteins are duly tagged, and as I told you, there is some disease that can due to the protein misfolding. Amyloid disease, accumulation of the insoluble spontaneous aggregation protein called amyloid, amyloid, for example, in Alzheimer disease, causes spontaneously by mutation in particular gene which they produce altered protein after abnormal proteolytic cleavage in some normal protein, a unique conformation state that leads to the formation of long fibrous protein. Yeah, that is some how to change some misfolding, some proteins they change, they make a disease. This just show you how this amyloid defect in the cerebral amyloid. Yes. Yeah, as I told you, this happening in the Alzheimer disease. So many studies going on now, how this unfolded or misfolding protein is happening, and they can affect the neurotoxic and central bastion, leading to connective impairment characteristic of the disease, and that is of the Alzheimer. You lost to memorize. Yes, take home message. Protein polymer consists of 20 kind of amino acid. Protein fold into a unique 3-dimensional. Protein has a hierarchical nature. Protein is closely related to its function. Protein misfolding leads to the disease. And we go next lecture will be about the protein digestion.